Hi everyone, this is Ms. Romani, and for this lesson we will learn about proteins, mostly focusing on their functions in living systems, their structure, and how the structure of proteins can actually affect their function. And proteins are usually very large molecules that are made of carbon, hydrogen, and oxygen, just like carbohydrates and lipids. But unlike most carbohydrates and lipids, proteins also contain lots of nitrogen, and depending on the protein, a little bit of sulfur. A 3D representation of a protein molecule will sometimes look something like this, which shows that a protein is actually made up of smaller molecules, shown here as little beads. These smaller building blocks of proteins are called amino acids. And if a protein is unraveled, it would look like a long string of different amino acids. This structure or formula here represents 3 different amino acids actually that are linked together. And proteins are extremely important molecules to living things. So important, as a matter of fact, that DNA, the molecule that codes for life, essentially contains only the instruction for making proteins in living cells. DNA does not code for carbohydrates. It does not code for fats. It only codes for proteins. That's how essential proteins are. Because if we have proteins, the proteins can take care of the rest. So here are some of the functions that proteins serve in living things. To start, proteins serve a structural function in living things, especially in animals. Animals are basically protein. Two clearly visible structures made of a protein called keratin are nails and hair. And internally, most of our organs, our bones, our tendons, ligaments, and pretty much most of our internal structures are made largely of protein. And of course, our muscles are made of proteins. If you or anyone you know works out regularly, you are aware that to build muscle, you need to eat a little bit more protein. And that's because what our muscles are made of is protein. More specifically, our muscles contain 2 proteins, actin and myosin, that interact with each other. And speaking of that, proteins also serve a contractile function in our cells. Essentially, any movement in our cells or by our body tissues happens only because of proteins. A perfect example of that is the movement of our muscles, and not just our biceps or our triceps, but the muscle contractions of our heart that keep us alive. They are all due to the contractile properties of actin and myosin proteins in our muscle cells. And it's not just muscle movement. Any movement that happens within cells is due to the action of contractile proteins. Like, for example, the spindle fibers that move the chromosomes during mitosis and meiosis. Or any movement of organelles within a cell. For example, vesicles are like little membrane packages that move material from one part of the cell to another or to and from the plasma membrane. But the way they actually move is not on their own. They move with the aid of little proteins called kinesin, which is depicted here walking along another set of protein fibers that make up the cytoskeleton, and they're carrying a vesicle to its destination. Those protein fibers of the cytoskeleton, by the way, also serve a structural function within cells. They anchor down all the organelles, and they give the cell some shape and structure. And cells themselves can move. Not all cells, but some of them do. Like, for example, sperm cells. They have this tail that is basically made up of a flagellum that help propel sperm cells towards the egg. And within the flagellum, there is proteins called microtubules that are similar to those found in spindle fibers. So shifting gears, proteins can also serve functions that are completely unrelated to structure and movement. One of their most important functions in the body is the role they play as enzymes, catalyzing almost every chemical reaction that occurs in living cells. Without proteins, cells will be unable to perform the chemical reactions that keep them alive. And most hormones are protein hormones. Hormones send messages to regulate our body processes. There are many different types of protein hormones, all with very different jobs. Insulin, for example, is a small protein hormone made up of only 51 amino acids. And this protein can help us regulate our blood sugar by acting as a sort of key that lets glucose from our blood into our cells. And proteins called neurotransmitters allow for communication via our nervous system. Essentially, nerve signals are electrochemical signals within each nerve, or as we call them, neurons. But in order to pass information from one neuron to another throughout our bodies or in our brains, or from one neuron to a muscle, our bodies use these proteins called neurotransmitters. You may have heard of some of these neurotransmitters. For example, adrenaline, also called epinephrine, which is released when we get scared or angry or excited, or dopamine and endorphins, which trigger sensations of pleasure or euphoria, depending on which one. And proteins also serve a very important role in transport, both within cells and within our bodies. Transport proteins embedded throughout our cell membranes are responsible for moving substances in and out of our cells. And also transport throughout our bodies occurs thanks to proteins. For example, our red blood cells are full of a protein called hemoglobin, and hemoglobin carries oxygen in our blood. And also our blood vessels themselves, our veins, our arteries, are made of protein. Well, they're made mostly of muscle, which is protein. And speaking of blood cells, our immune system cells, often called white blood cells, produce antibodies that defend us against diseases. Antibodies are tiny little proteins that look a little bit like the letter Y, and they attach to pathogens like viruses and bacteria in order for our immune system to recognize them and then better fight them. And that's not all. Proteins in our blood form platelets and clotting factors that stop us from bleeding out and help us heal cuts. They make up a skin protein called collagen that gives our skin elasticity, and which unfortunately falls apart as we age, which is why we get wrinkles. They are pigments like melanin that protects us from UV radiation and also darken our skin, our hair, and our eyes. They can even be used by our cells for energy. If we run out of sugars and fats, our bodies can actually turn to protein as a source of energy. And these are just a few of the roles that protein play in our bodies, our human bodies. If we look at other living things, we will find a much larger variety, including, say, for example, fluorescent proteins that make some organisms glow in the dark. So as you may have gathered, proteins serve many extremely varied functions in our cells, in our tissues, and the cells and tissues of many living things. And the reason for this variety has to do with their structure. So let's explore protein structure. As mentioned earlier, proteins are made of monomers called amino acids that are linked together. Each amino acid has a common general structure. So we can start with the central carbon. And like all carbons, it forms 4 bonds. One of them is to a hydrogen, another is to an amino group, and a third is to a carboxyl group. And then our final bond is to an R group. Now, our lesson on functional groups never mentioned R groups, and that's because an R group is not a functional group. It is a placeholder, or a variable like X in algebra. And in the case of amino acids, there are 20 different side chains, all with different properties. So let's explore now how amino acids can combine to form the vast variety of proteins that we see on Earth. To start, amino acids must be bonded to each other via dehydration synthesis reactions. Now, like always, a dehydration synthesis reaction will remove water from the molecule to form a bond. And like always, the reaction will occur between the functional groups in the molecules. So in the case of proteins, the bond will form between the carboxyl group of one amino acid and the amino group of a second amino acid. The new bond formed between the 2 amino acids is called a peptide bond, and the 2 amino acids bonded together are called a dipeptide. Let's also not forget that just like with carbohydrates and lipids, the bonds formed between the monomers of proteins are broken by hydrolysis reactions and the addition of water into the bond. Now, if a dipeptide is 2 amino acids bonded together, then many amino acids bonded together form a polypeptide. A protein is actually a polypeptide that is folded into a very specific 3-dimensional shape, specific to the function of the protein. Because just like everywhere in life, when it comes to proteins, structure determines function. And for proteins, the structure is ultimately determined by the amino acids that are linked together in the polypeptide chain. For example, this is what the polypeptide for the human version of the hormone insulin looks like. Insulin is a very small protein made up of two polypeptide chains attached to each other. One of them, chain A, consists of 21 amino acids, and the other, chain B, of 30 amino acids. But it's not just any random amino acids. The specific amino acids that make up both polypeptide chains in insulin determine how insulin folds into a three-dimensional shape and how it can interact with cells. Protein structure can be described using a hierarchy of 4 levels called primary, secondary, tertiary, and quaternary structures. The primary structure of a protein is simply the sequence of amino acids in a polypeptide chain. This sequence is determined by the genetic code in DNA. When we look at the primary structure, we don't pay attention to the shape of the molecule, only which amino acids are found linked together and in what order. In most proteins, parts of the polypeptide chain are coiled or folded, forming twisted helixes or sheets. When we describe the secondary structure, we're describing these localized conformations that are happening in small sections of the protein and involve only a few dozen amino acids in the chain. One type of secondary structure is called the alpha helix that happens when the backbone forms a spiral. Another is called the beta-pleated sheet. And in this structure, the polypeptide chain will extend one way, then fold back and forth a few times, forming parallel lines. Now, one thing that you need to know about secondary structures is that they're held in place by hydrogen bonds. And these hydrogen bonds form between the atoms of the backbone of the polypeptide. What this means is simply that the R groups are not involved in this level of structure. When we describe the tertiary structure of a protein, we're describing how it folds into a three-dimensional shape. This folding is very specific to each protein because it is the shape that determines the protein's function. And it is determined by several different interactions that occur between the R groups that make up each polypeptide. Now, most proteins are made up of a single polypeptide chain that is folded into a three-dimensional shape and only have the 3 levels that we've just discussed. However, some proteins are made up of multiple polypeptide chains, also known as subunits. And when these subunits come together, they give the protein its quaternary structure. And one example of a protein with a quaternary structure is hemoglobin. Hemoglobin carries oxygen in the blood and is actually made up of 4 separate polypeptide subunits that come together to form this final structure. And so there you go, the 4 levels of protein structure. The primary structure that is held together by peptide bonds between amino acids, and the secondary structure, which is stabilized by hydrogen bonding between polar functional groups in the backbone but does not involve the R groups. The tertiary structure that folds the protein into three-dimensional shape determined by various bonding and interactions between different R groups. And the quaternary structure that attaches 2 or more folded polypeptides into a larger protein. Now let's take a look at what happens when we change the structure of a protein, or at least let's take a look at what could cause a structure to change. When you cook an egg, for example, the protein albumin that is found in the egg white loses its natural shape, which is why a cooked egg looks and feels different than a raw egg. This process by which a protein loses its three-dimensional shape is called denaturation. So heat can denature proteins, which is what happens during cooking. But heat is not the only thing that can do this. Radiation, acids and bases, and various solvents, heavy metals, like for example lead or mercury, and really a lot of different chemicals, all can disrupt the tertiary and the secondary structures of proteins. When these bonds and interactions are disrupted, the protein will lose its three-dimensional shape and will turn back into an unstructured and non-functional string of amino acids or an unshaped polypeptide. And a lot of the time, denaturation is permanent, like when cooking an egg. That's irreversible. It will not return to its original raw egg state. But researchers actually have found that some proteins can refold on their own right after denaturation. Since these proteins can pretty much go from unstructured to folded all by themselves, it is the amino acid sequence then that must contain all the information needed for folding. So just to recap, denaturation is about the loss of that secondary and tertiary structure, but not the primary structure. The peptide bonds between amino acids are still there, but the protein just becomes unfolded. And when a protein is denatured, when it becomes unfolded, it loses its shape, and therefore it loses its capabilities of performing its original function. And that's it for this lesson on proteins. Talk to you later.